Thank you very much, Alison, for agreeing to give a first Build a Cell seminar of 2024. And it's all yours.
Great. Thank you very much, Kate. So Happy New Year, everybody. My name's Alison Smith, and I'm from the Department of Plant Sciences at the University of Cambridge in the UK. And I want to tell you about the project that I've been working on for about 4.5 years with my colleague Saul Purton, who is in the Department of Structural Molecular Biology at UCL, University College London, also in the UK. And the project is towards the Chlamydomonas chloroplast genome version 2.0 or 2.0. We slightly have an argument about whether it should be, we're trying to have a synthetic version of it. So I don't know whether the synthetic version should be version 1.0 in the first place or 2.0, but anyway, because there is one already, that's what I mean. Okay, so what I'd like to do is to say, well, why do you want to redesign it? Why do you want to build it? Talk about the design principles and methodology that we've been adopting. And then tell you something about what we've been doing in terms of deletions, refactoring, and recoding. And then the options for, once we've done the, understood the plastome itself, how we might be able to exploit the synthetic plastomes that we can prepare. So why design a genome? Well, this is the sort of holy grail of synthetic biology. If you deconstruct something and then put it together and it works, then that gives you a much better idea about how the thing functions. And so from a genome's perspective, it tells you about the minimum requirements. I'm just trying to get rid of the bit at the top. Okay.
We don't see, Alison, we don't see your bit on the top. I think it's just Zoom displaying stuff.
I know, but I can't see my slides. I've got rid of it now. So it tells you something about the minimum requirements for a genome. What is it that you need? How can a genome initiate? And the ability to build it from constituent parts means that you can redesign it and you can modify it so you can exploit it for your own particular ends. And this sort of approach was built out of an initiative that was in the UK, run by my colleague Jim Hasselhoff, together with Anne Osborne at the John Innes Centre in Norwich, called Open Plant, which is a synthetic biology centre based on plants. And the whole ethos of this was to try to understand the principles of how plants are built, in which they also include algae, and to make it open so that all of the knowledge is shared. And we can go through this virtuous design, build, test, learn cycle, and everybody will learn from it. So this, as I said, the sort of holy grail is to build a minimum genome. And the idea is has been sort of pioneered perhaps by many people, but Craig Venter in particular. And his institute was probably the first to build a synthetic genome of a bacterium. And this was from Mycoplasma mycoides. And the first version of the genome, which was called SYN 1.0, was about 1 megabase or 1 million bases. And then through 4 additional design, build, test, learn cycles, they ended up with SYN 3.0, which was about 500,000 base pairs, so 500,000 base pairs. It had 473 genes, and this was enough to run the cell once it was introduced and the original genome was removed. But there was still about 1/3 of the number of genes were of unknown function. And that's the problem. You can't really test something like this unless it works in the cell. And so there could be parts of that sequence which are not necessary. And if you don't really know what they're doing, you can't then redesign it effectively. So we wanted to take a system that was much, much simpler. And our focus has been on chloroplast genomes. So plants and algae have chloroplasts, and they all have these semi-autonomous genomes present in about 40 to 90 copies per chloroplast. And this is showing the genome map from Chlamydomonas, a unicellular green alga that I work with. And actually, there's only one chloroplast in the Chlamydomonas cell. So that's why the same number of copies in the cell. The size of the genome, the chloroplast genome, is actually very, very similar in most organisms. It's between 120 and 200 kilobase pairs. So Chlamydomonas, or Chlammy as it's abbreviated to, is one of the largest at 200 kilobases. But all these genomes contain roughly the same number of genes, about 100 genes, of which 70 or so are protein coding. And then there are genes for the ribosomal RNAs and the tRNAs. And in the vast majority, but not all genomes, chloroplast genomes, the ribosomal RNAs, which are shown here in red, are duplicated in this inverted repeat. So, sorry, I should get my laser pointer. So in this inverted repeat here, so you can see this region here is duplicated on the other side, but in opposite orientation. And so the reason for choosing the chloroplast genome is because it's so much simpler. It's semi-autonomous, which means that if you have a mutation in the genome, you don't necessarily kill the cell because the nuclear genome is still functioning. And most importantly, you can do simple transformation via homologous recombination. So you can precisely introduce sequences into the genome and knock other ones out. Now, the Chlamydomonas plastome or chloroplast genome is extremely well characterized. There's a single RNA polymerase, which is encoded, the 4 subunits are encoded by the genome itself, and there's one sigma factor, which is encoded by the nucleus. In higher plants, there are multiple RNA polymerases and sigma factors. So it's much simpler, this genome. Plant chloroplast genomes are characterized by having gene editing. There's none of that in the Chlamydomonas chloroplast. And the nucleus of Chlamydomonas exhibits silencing, post-transcriptional gene silencing, RNAi mediated, and there's no silencing in the chloroplast. So in terms of trying to manipulate it and then redesign it, it's sort of virtually the most simple system that you could imagine. Further advantages of this system for the chloroplast redesign is that the photosynthetic genes, which are the ones shown in green, and they are principally for photosystem complexes, photosystem 1 and 2, the cytochrome complex, and the ATP synthase. These are all dispensable. So they have already, in previous studies, been knocked out individually, and the cell is functional because you can give it a carbon source. It doesn't require photosynthesis to grow. It actually grows on acetate. In addition, about 7 or so years ago, all of the genes for photosystem 2, PS2, were knocked out and they were refactored. So a new set of genes clustered together were inserted back into the genome, and that restored photosynthesis. And there has been one report that the complete genome has been synthesized, assembled in yeast, and then reintroduced. But because it was the wild type genome, it was subject to considerable recombination because of the very active homologous recombination in the chloroplast. And as a result, a chimeric molecule was obtained. So it wasn't really showing what the minimum genome could be. Nonetheless, based on all these factors, we decided that this would be an ideal system to try to deconstruct and then redesign completely de novo. So what we've been working on is sort of 3 different approaches. We've been doing minimization, that is deleting all of the dispensable regions, which would enable us to identify the minimum plastome size. We've been refactoring, which is reorganizing the genes into functional clusters. This will facilitate engineering, but also show if there are any sequences that we can't identify, which are important for normal function, and recoding, that is reassignment of the genetic code to reduce complexity. And using the information and the results from these three approaches has enabled us to consider how we might go about redesigning the plastome completely. In order to be able to do this, we needed to establish the methodology. So we needed a rapid method for construct assembly. We had to optimize transformation. And because there are multiple copies of the chloroplast genome in the chloroplast, the transformation and the homologous recombination is almost certainly going to happen in a single molecule. And then we have to select for homoplasmy so that all molecules have now got the modification. Because we wanted to do iterative processes, we wanted to knock out one gene, then another, and so on, we needed to be able to recycle the selectable markers. And we needed to have ways to test the phenotype of our engineered chromosomes to assess whether it had any effect on the function of the cell. So we set this up, and I'm going to show you a couple of examples where we've used the approaches to test which genes are essential. So I said that all of the photosynthetic genes have been knocked out and the cell is viable as long as you give it acetate. There are other genes which are essential, and they're to do with the genetic machinery, the ribosomal machinery and RNAs. But there are some enigmatic open reading frames whose functions are not necessarily known. And in Chlamydomonas, there are these two very enigmatic genes called Wendy1 and Wendy2, and they're present at around 12 o'clock here and at about 8 o'clock here. So what we wanted to, the function is completely unknown. Single knockouts are viable. And we wanted to be able to see if a double knockout was viable. But in order to be able to do that, we had to recycle the marker. So we've established this method here, which is called positive-negative selection, or POSNEG. And this involves the introduction of a selectable marker, which is a bifunctional protein. So in the schematic below, this is the endogenous chromosome, and then this is the plasmid that we're going to introduce. And then these two regions, which are in the green shaded boxes, are the homologous regions, which we have present in this plasmid, which enables homologous recombination to take place. And within these regions, we've inserted luciferase, which is a reporter enzyme. And this is what we use to measure the efficiency of translation, or transcription and translation, in our modified strains. And then this is the selectable marker, which is bifunctional protein, fusion protein, with AADA, which confers resistance to spectinomycin, and COD-A, which is confers sensitivity to an antimetabolite, 5-fluorocytosine, or 5-FC. So if we introduce that into the genome, that we have, in addition, these two sequences either side that flank this selectable marker. We can select on spectinomycin, so we know that we've got the construct in the genome. But then we can counter-select on 5-FC, and this cells that still encode COD-A will die. And so there is only if they can loop out this sequence here that they will survive, because they no longer have the COD-A gene. And these two sequences are in direct orientation, so homologous recombination between those will actually excise this whole sequence. This works really very well. What we wanted to do was to establish what the length of the direct repeats is needed in order for this to happen effectively or efficiently. If there are no direct repeats, then we get insertion of the selection marker, but we don't get any loop out at all. If we have sequences that are 200 300 or 400 bases, then we do get loop out. But as the length increases, it's much more efficient. And the way we test for this is doing PCR using primers across the different sequences. And you can see here, this is the presence of the selectable marker. And so we have, we still have a selectable marker here in these, in some of the strains where we had a 200 base pair direct repeat. If we've got loop out, then we can see the appropriate sized band, and then we can detect whether Wendy2 is there or not. This to illustrate the efficiency of the homologous recombination, we actually see spontaneous loop outs even without the selection of 5FC. And this is actually one of the hurdles we have to overcome, because we can get homologous recombination with homologous sequences when we don't really want them. So we have to be careful in using the various parts that we are using in our constructs not to have too much duplication. Otherwise, we get these spontaneous loop outs and aberrant genomes. Okay, so we've managed to generate a Wendy2 knockout in a Wendy1 knockout. And so we've got both of them present, and we decided to assess how well they grew. So this is growing them in different media. HSM is photosynthetic media, and so it requires the photosynthetic apparatus for the cells to grow. And TAP is the media which contains acetate. And the TAP media, which is shown in circles, they both grow. So this is the wild type in green and the double knockout in blue. They both grow essentially the same. The squares are the ones in TAP in the dark, and then the triangles are the ones, sorry, the diamonds are the ones in HSM in the light, and then if in the dark without acetate, they don't grow at all. So there's basically no difference between those two things. And this is the phenotype that we use to test whether the genes that we've knocked out are important or not. So we've used this approach to knock out regions all the way around the genome. And you can see that they vary in size. So this is the Wendy2 gene, for example, is 2.3 kilobases. Some of the larger sequences we've knocked out are 8.7 kilobases here, and 22 kilobases here, where we've knocked out the inverted repeat. Come back to that in a minute. So, so what we have also done is to, because we can recycle the marker, is that we knocked out one region. So for example, we've knocked out this region here, and then we've knocked out another region as well in the genome. But the challenge here is that when we've knocked out the first region, the cells are non-photosynthetic. So, and they're also light sensitive. And that's actually quite a challenge to get sufficient biomass to do efficient transformation. But using a number of sort of physiological tricks, we've managed to establish the means to do that reasonably effectively. So as I've mentioned, we wanted to see the largest size that we could knock out using this approach, and we targeted the inverted repeat. These are 22 kilobase pair sequences in opposite orientation, and this actually allows recombination between them, so that you can recombine, and then the so-called small single copy and large single copy regions swapped around relative to one another. And these, the sequences which are repeated contain the ribosomal RNAs, 2 of the tRNAs, and one of the subunits of photosystem 2. So because we've got a mixed population within the in the chloroplast, because you'll have it's assumed that there's equal proportions of both these types of molecules. There are 4 possible deletions that you could do. You could delete this region, you could delete this region, this one, or this one. As it turned out, we only managed to delete 2 of the regions, and we didn't find any transformants that had that would deleted this repeat or this repeat. We're not sure of the reason for that. It actually had to it took place over 2 rounds of transformation, because the first insertion was an insertion rather than a knockout, and then we did the counter selection and then got the deletion. So it was rather long-winded, and Pavel Darker, the postdoc who was doing this, wasn't keen on repeating the experiment to see what happens. Nonetheless, we had these 2 strains which have got 1 of the repeats knocked out. And this was confirmed, the homoplasmy was confirmed by nanopore sequencing. We don't see any of the parental genomes present in our chloroplasts. If we look at their autotrophic growth, so this is in HSM in the light, there's possibly a slight difference, so that this version is slightly lower growth rate and carrying capacity than the parental strain, which it's not the wild type directly. But it's not really, I wouldn't say it was statistically significant. Well, that's statistically significant. And then using luciferase as our reporter for the genetic machinery, we don't really see any activity difference. And this is, I should say, because in the parental strain, the luciferase gene is in the inverted repeat. So in the knockouts, where we've removed, sorry, in the deletion strains, where we've removed 1 of the inverted repeats, we've reduced the copy number of the cassette. And that would probably account for the difference in activity that we see. So that was very encouraging, that we could knock out this very large region and still have normal functioning of the genetic machinery. We then wanted to extend it to see whether we could knock out regions in one go that contained functional genes. So these 2 genes here, YCF1 and RPOC1, these are essential genes shown in black. And then in addition, there are these 4 tRNA genes for histidine, methionine, phenylalanine, and asparagine, which are also in black. So they're also essential genes. And they're interspersed with these other sequences, which are not essential. So for PSAC, that's a subunit photosystem 1, PSBC, a subunit photosystem 2, and PET-L, which is a subunit of the cytochrome complex. And there's also this metabolic enzyme, OR528. So we wanted to see whether it would be possible to knock out this whole region, thus deleting the ability for 3 photosynthetic complexes to function. And then that would then facilitate refactoring the genes for those photosystems in the next step. But we had to consider what we could do with the tRNA genes. So we could have simply added those genes to our cassette. We could have put it in with a selectable marker plus the tRNA gene. So we're basically knocking out the whole of this region, but we've been reinserting the tRNA genes back into this position. So that's one possibility. But that's essential for the genes. But the other possibility was actually to relocate those tRNA genes straight away. So here is a deletion locus shown on the genome. This is the bit that we want to knock out. And there's another locus which also has 4 tRNA genes on the opposite side of the single copy region. So we could see whether we could transfer those 4 genes from here to here. We'd end up with 8 tRNA genes. And then this would also facilitate refactoring and inserting these photosynthetic genes in another neutral site later on. So this is what Pavel did. This is the new acceptor locus, which has got these 4 tRNA genes, just shown now with a single letter code for the amino acid. And we inserted into this sequence this cassette here. This is the selectable marker. Here are the 4 genes that we want to translocate, but actually they're in a different order to the order in the original locus to try to prevent homologous recombination in that region. And we also duplicated N and F genes upstream of the selectable marker, which should then facilitate loop outs. So this region here is homologous to this region here. And by looping that out, we would end up with, we would remove the selectable marker. And this was successful. And it's worth pointing out that here, in this case, we've actually got 2 copies of these 2 tRNA genes in this locus and in the donor locus. We've actually got 3 copies of the N and F tRNA genes. Here, we've now just got 2 copies of those 4 tRNAs. It then enabled us to try to knock out the entire cluster, and that was successful as well. So this is strain 157, which has got the duplicated tRNA. Sorry, I should say this is the strain 157, which has got the duplicated tRNAs. And then strain 289 has got the large deletion with the photosynthetic genes knocked out. So we'll only grow in the dark, well, I only grow heterotrophically with a source of acetate. If we look at strain CSB 157, so this is the one with the duplicated tRNAs, it shows normal growth under several different conditions. So the growth measured here is identical to that of the wild type strain. You can see in the dark, although it does grow, it then sort of peaks and tails off. And this is characteristic of all strains in the dark because light is really needed for normal functioning of many of the cellular processes. So it can keep going, but it's not very happy. In the tRNA refactored strain, so now it's got the deletion of the, with the deletion of those photosynthetic genes, you can see that it essentially grows the same as the strain that's got the refactored tRNAs, but still has the photosynthetic genes. And this is a feature of the knockouts and so on that we produce, is that basically they don't really differ from any of the, from each other in their physiology. So then the last thing I want to tell you about that we've been doing to try to understand what's required for the function of the genome is to alter the genetic code. And this has been pioneered by Jason Chin's group in E. coli. So the idea of changing the codons in a genome to see how well they can function with fewer codons, and then perhaps to introduce codons which code for different amino acids. So what they did in 2019 was to take the E. coli genome, or to generate completely synthetic E. coli genome that used just 61 codons instead of 64. In order to do that, they eliminated codons for 2 of the serines. So serine has 6 codons normally, and they also eliminated a stop codon. This required them to design and synthesize and build a genome that had 18,214 different changes. So it was a big challenge. The Chlamydomonas plastome that now comes into its own because it's really so much simpler. So this is a codon usage table for the Chlamydomonas plastome. And you can see each one of these boxes is one set of 4 codons. And I'm just showing you here, I'm just pointing out the stop codons. So the stop codons are UAA, UAG, and UGA. there are no genes that use UGA. So it's possible to eliminate that straight away. The vast majority use UAA, and 4 of the genes use UAG. So this is much, much simpler. And my colleague Saul has demonstrated the use of, or the substitution of UGA codon for a modified amino acid. So what Pavel, so this can be reassigned without affecting any endogenous genes. These would obviously affect the endogenous genes unless you altered those. So what Pavel did was to take advantage of these observations and come up with a codon compression strategy that enabled, in principle, the Chlamydomonas genome to be reduced to just 51 codons out of the 64. So just as an illustration, these are the codons for leucine, and there are 6 altogether. And in the Chlamydomonas chloroplast, there are 2 tRNAs, one that recognizes these two codons and one that recognizes these 4. So in principle, we could eliminate all 4 of these codons and just use these 2 codons, and it would still incorporate leucine at the appropriate point in the protein. And in addition, you'd also lose 1 tRNA. Taking this approach, he was able to work out which other codons that you could lose. So 1 of the isoleucine codons, 2 of the arginines, 2 of the serine, 1 of the glycine, and 2 stop codons. So in order to end up with a genome that had just 51 codons, it would require changes in 2,000 codons, so many fewer than E. coli. And this would be about 7.5% of the entire genome. So that's the strategy. But the question is, would it work? So we decided to try to do 1 gene at a time and test the effectiveness of that. And the gene that Pavel chose was the RPOA gene. So this is for the alpha subunit of the RNA polymerase, which is encoded by the chloroplast, as I mentioned before. So there were, using the strategy that I've just shown you, there were 55 codons out of the total that could be altered, shown here, so 19 leucine and so on, which is about 7.5% of the codons within the genome, within the gene. So what he did was he designed a sequence where the black lines show the positions of these codons which have been altered to be the redundant ones. In addition, in the particular design, he incorporated a flag tag into the protein because RPOA had been shown to be modifiable by this. And we wanted to make sure that if we didn't get normal function, we could still see whether the protein was being produced. In addition to the simple manual compression, Pavel used 2 programs which had been proposed to facilitate randomization of codons within the within any one gene. One was called Chimera map, one was called CUO. And the reason for doing that was to detect the tolerance of the changing the codon so radically within the chloroplast, and also to try to minimize homologous recombination to the maximum extent. And what he was able to do was to get homoplasmy for 2 of those 3 designs. So the manual one, he could show integration and no presence of the wild type by using PCR. And also for the CUO design, but not for the Chimera. So it always retained a wild type version of the genome present in the cells, suggesting that there was something in the design which was not tolerated as a functional or for a functional cell. I should have made this clear. RPOA is obviously an essential enzyme because you need RNA polymerase for the normal genetic machinery of the chloroplast. So the chimera didn't work, but the other two did. And then using our two methods for testing the phenotype, this is the phototrophic growth, so this is an HSM. There's no difference between the control and so this is the wild type. This is the wild type containing luciferase marker, and then the two modified recoded strains. And then this is looking at the activity of the LuckCP reporter in these different strains. And again, there isn't any difference between the activity. So no effect on the genetic machinery, RPOA obviously being required for transcription. The next thing he tried was to integrate or to recode a large operon. So again, just to orientate you, this is looking at the chloroplast genome here, and the black genes show the ones that are essential for function. So this is another subunit of the RNA polymerase. And then in addition, we have these ribosomal proteins, RPS9 and RPS18, and then RPS2. So these are all essential. And then this is RPOA as well. So this is taking the strain that we'd already modified, so it had already been recoded. Pavel introduced this entire modified region where it's all been recoded. And he got in 80% of the transformants, there was integration of the full-length operon, and 95% of them showed homoplasmy. So in the few that didn't is probably simply not long enough to sort out. Now, one additional trick that he's used in order to facilitate integration of such a large sequence is to have a selectable marker at one end and the other. So in addition to our COD-A AAD marker, he put in another antibiotic resistance gene at the other end, which supports the integration of this quite large sequence, of 8.5 kilobases. Now, the recoding was confirmed by targeted next generation sequencing. You can see this is just the reads, and you can see the vast majority of them are really 100% the recoded sequence, with only a few aberrant or original wild type sequences present. We actually got very similar results with the recoding of a non-essential gene for the enzyme Ribisco, but it's a very, very highly expressed gene. And again, we saw efficient homoplasmic replacement. And in addition, that it was also, there was no change in growth of the cells nor in luciferase expression. So what we've managed to do is to establish a methodology that enables us to do minimization of the plastome. And this has enabled us to identify the dispensable regions so that we're pretty certain we know what's needed and what's not needed. We've done some plastome refactoring with the tRNA genes. We have started with the photosynthetic complexes, but haven't got the data to show you that yet. And we've done very extensive plastome recoding with several genes that have been expressed normally using just 51 codons. So we're pretty confident that we can use this approach, which only takes 4 to 6 weeks to generate the transformants, and also get rid of the selectable marker. And in all of the cases, we don't see any significant impairment in function. And as a consequence, we think we now have a design for our synplastome version 1.0 where there's no inverted repeat. We've deleted all of the non-essential genes, but we're not going to delete the photosynthetic genes in the first version because, as I've said before, the strains grow much more poorly and therefore they're difficult subsequently to modify and handle. But we are going to delete the all of the other regions that we've identified as not being essential. There are a number of small direct repeats in the genome known as SDRs. We're going to delete Wendy1 and Wendy2. There are some introns in the genome, and we're going to remove those. There's even some there's a trans-spliced gene, so it's present in 2 parts on the genome, and we're going to combine them into a single gene. And it will have a 51 codon genetic code. So the point that we are at the moment is we've done the design, and we're now trying to, we're now ordering the parts. The last hurdle we have to overcome is the fact that the genome is very AT rich. It's in some regions it can be 35% GC or 65% AT, and that's actually quite difficult to synthesize. So we are having to do it in quite small fragments, and then we will assemble them. But we hope in the next 3 months or so to have all the parts, and we will be able to assemble them in yeast. Then we have to introduce them into Chlamydomonas. That's a whole new, the whole genome. That's a whole new set of challenges. I don't have time to tell you about those. But we do have a number of potential ways to try to ensure we don't get a nasty chimeric mess, as in the previous experiment. So once we've got our genome, what are we going to do with it? Well, I think we do want to end up with the real minimal genome. So I should say that that's about 168 kilobases. So we'll have lost about 35 kilobases. But as I've said, we're going to keep all the photosynthetic genes. Once we've shown that we can do this with this genome, the next obvious thing is to build a genome without any of the photosynthetic genes. And that would be about 110 kilobases. So that would really be a very minimal genome. Then we have a chassis that we can use for the production of high-value chemicals. So we're interested in making high-value chemicals like terpenoids, which are potential therapeutics, vitamins, carotenoids, recombinant proteins, and so on. We will have what's known as a naive chassis that will facilitate the optimization of the reduction of these compounds. Together with colleagues at University of Edinburgh, we're interested in improving photosynthesis, and we will do that by trying to introduce the refactored photosynthetic genes, but maybe using genes from different sources, and in addition, novel ways for carbon fixation to try to overcome issues with the oxygenase activity of ribisco. We're interested in introducing novel functionalities, including the genes for nitrogenase, which would facilitate or allow nitrogen fixation, or other pigments that would extend the spectrum of light capture. And then using the recoded genome, we really want to go beyond the genetic code that we're, the universal genetic code, and try reassigning the tRNAs so that they can then accept different amino acids. And so this is shown here in this further scheme that Pavel established. So I explained this already, but by removing these 4 codons, we've now got, we only need 1 of the tRNAs. Then we could reassign that tRNA to insert an amino acid a functional amino acid, such as phenylalanine. And then we would actually remove this set of codons, so we could compress further. And then delete our redundant tRNAs. But in addition, we can then introduce these tRNAs along with their amino acyl tRNA synthetases, which accept non-canonical amino acids. And then this would enable us to produce new to nature proteins, or neoproteins as they're known, where we've got non-protein amino acids incorporated via translation into these proteins. And there's enormous interest in doing this, both from a fundamental understanding of proteins, but also for the potential to have novel catalytic activities for industrial purposes. Okay, so I hope I've given you a flavor of the sort of things that we do in our lab. I'd just like to say that I didn't do any of it, but all of the experiments have been done by Pavel in my lab, along with his technician, Kitty Clouston. And this is a joint project with Saul at UCL, together with his two postdocs, Harry and Henry. And thank you very much for listening.
Thank you. Thank you so much, Alison. There is a lot of questions in chat. Can you see chat?
I can if I get rid of my laser pointer. Hang on.
If you could read a question out loud before answering, that's going to help people watching the recording.
Yeah. Okay. So Avery says, do we know all the genes that plastids outsource to the nuclear genome? Genes necessary for plastid growth and division that are now encoded in the nuclear genome? Yes, we do. There are some organisms which have very reduced plastid genomes, and all of the sort of and they tend not to be photosynthetic. And so all of those genes have been transferred to the nucleus. So the vast majority of the genes that are needed for chloroplast function, there's probably about 3,000 proteins in a chloroplast. And there's only 100, well, there's only 70 or so protein coding genes. So all of the basically all of the metabolic enzymes, all of the enzymes for lipid biosynthesis and membrane assembly and so on are all encoded by the nucleus. I hope that's what Avery was asking. You can, I don't know if you can, they can
unmute themselves if they want to.
I can stop sharing actually and stop staring at myself, and then Avery says again, sorry if I missed it. When you are recoding the whole operon, do you account for the change in tRNA usage, changing tRNA expression levels, or is it not needed? Well, that's a very good question. We didn't know what the result of that would be, but we don't see any difference in growth when there are, say, 3 copies of the tRNAs or 2 copies of the tRNAs. So yeah, I don't think it's necessary that we had to do that. I mean, it might be at the sort of granular level, but at the broad level that we're just trying to analyze at the moment, which is, how well do they grow and how well do they make luciferase as a reporter protein? There's no difference. And as a potentially nutty follow-up to Avery's first question, this is from Maya. Can they be reintroduced as chloroplast expressed only, either through nuclear genome knockout or interference with import machinery? Or are there cytoplasmic post-translational modifications that would make that possible? That's a very, very good question. It has been addressed in other systems and in Chlamydomonas. So we have introduced nuclear genes into the chloroplast, which are, it's not my work, so I won't go into details, but we have introduced some nuclear genes that were in the nucleus into the chloroplast, and the cells function fine. So we've knocked them out from the nucleus and then we put them in the chloroplast. Actually, more people have, or there's been much more work doing it the other way around, which is taking the chloroplast genes and putting them in the nucleus and getting rid of them from the chloroplast, because originally chloroplasts arose as endosymbiosis of cyanobacteria, which would have had the 3,000 genes in them. And then they lost all of the genes to the nucleus. So the question is, why are there any in the chloroplast to start with? Or not to start with, or now? Why are there still some left? And probably there isn't any need for them to remain in the chloroplast. You can move them to the nucleus. So it's probably just a snapshot in evolutionary time that we're at the moment. So the cytoplasmic post-translational modifications, there is no evidence of glycosylation of chloroplast proteins. There is some processing, which might occur during the import, but all of the genes that have been introduced back into the chloroplast are the mature protein. So after any processing, we haven't tried to put in the protein without making those modifications. So I'm not certain whether that would be necessary. Okay, shall I just carry on going? I feel like I've been
Yes,
talking
please.
forever. I'll just take some water if I may. So Arga says, could you please clarify one thing? You mentioned Rubisco as non-essential. I thought it is crucial in photosynthesis. Yes, you're right. It is crucial in photosynthesis, but Chlamydomonas does not need to have photosynthesis to grow. So we can knock out the photosynthetic genes, including Rubisco, and as long as we give them a source of fixed carbon, which is acetate, they will grow. So I hope that's clear now. Zanar says, do you plan to study how the genome redesign affects genome stability? Yes, we're in the process of doing that. Again, well, so the genome is found in nucleoids, which are also found in bacteria, and it's associated. These nucleoids are generally associated with the thylakoid membranes, the photosynthetic membranes, and you can visualize them as sort of punctate regions within the chloroplast if you use DAPI staining, for example. We have some preliminary evidence that in the inverted repeat deletion strain, there are fewer nucleoids. But over time, and we haven't had these strains long enough to know over years whether it would be stable, but certainly over months, we still see the genome present in its in the form that we've engineered it as, even though the number of nucleoids are reduced, and the copy number is also not altered. So even though in that case it's 22 kilobases smaller, it doesn't appear to have affected the stability. One thing that I should explain is that although I show the genome as circular, it maps as a circular genome, and you can sequence it, and there's a single circular molecule. Probably, well, I don't know about it. I'm not certain if they've done it in Chlamydomonas, but in many other chloroplasts, it's actually present in concatenated linear sequences. So they're not actually existing as circles. So the length of it is less of an issue, let's say, than a circular bacterial chromosome. Okay, Maya says, have you been able to reinforce the tRNA codon anticodon stringency? Yeah, so we reassign CTT and CTC separately from CTA and CTG. Yeah. Do you have to change the translation machinery to make sure the last base is distinguishable? We haven't had to do that. The reason that we have done that reassignment is because of the very high AT rich nature of the genome. So the predominant codons are the ones that end in T or A. So that's where that's the codon bias of the genome. So that's why we were doing it separately. And I'm not certain whether we've contemplated trying to recode it using the least favorable codons. I'm not certain that yeah, I don't think we've thought about that. So maybe that's worth thinking about. Thank you for that suggestion. Okay, so Maya preempted, she asked, is it purely academic interest in recoding, or is it for making non-canonical amino acids? Well, the answer is yes, as I said at the end. And then Emma Jane asks, will you be transforming whole engineered genomes into the chloroplast, or will you replace it portions sequentially? If you choose to transform whole genomes, are there any techniques to avoid homologous recombination with the wild-type genome? Okay, so yes, we are going to try and do the whole genome. We are not going to do sequential replacements. That's been there, done that. And our point is, can we build a whole genome and then introduce it? Now, there is this issue of homologous recombination, which I've mentioned, which would confound it if it was similar enough to the host genome. So what we have in our design is we've tried not to have any sequence longer than 200 base pairs where it's completely identical. It's not possible to do that completely, but overall, certainly in all the coding sequences, we don't have anything that long. There are certain intergenic spaces which we need to, which are important for gene expression, and we've had to retain them. And yeah, so I think, yeah, we're going to try and build the chromosome in yeast and then try and transform the whole thing into the chloroplast. There are lots of things that we'll need to do to try to avoid the homologous recombination. And it involves things like having a Rec-A minus a host strain, recipient strain. We will reduce the number of endogenous chloroplast molecules in the host strain before we introduce our redesigned one, and so on. But yeah, that's our next challenge. I think
you answered.
That's it. Yeah.
Does anyone want to ask a question with their mouth rather than chat?
Hi, Bob.
Hi, really exciting talk. I get super excited about this. So, you're choosing this genome that is not the smallest chloroplast genome, and the reasons are obvious because of the tools that existed. But at this point, do you think it's worth it to go for something small? And I'm asking because we're working with this 117 diatom genome that we can now clone in yeast very nicely. So, would it be worth it to try them both at this time?
Well, we started this a while ago before routine chloroplast transformation in diatoms was possible. But in addition, it's not trivial doing this stuff. As I said, I didn't do any of it. It was all done by the postdocs. But actually, it's the decades of experience of stuff that I've done and also Saul has done that I think makes it possible to overcome some of the challenges. It's hard to, there's a sort of I spent some time in France on learning techniques there, and it's a thing in French which is called a coup de main, or a green fingers, you'd say in English. I don't know what the equivalent would be in Hungarian, but anyway, something that is like, how is it that you can get something to work? And there's something mysterious about all of these things. It sounds a bit ridiculous, why, when we're trying to design something, but actually you have to have a live cell at the end of the transformation. And if everything is against you and it just about works when you're doing something very straightforward, when you're trying to do something that's not straightforward, like multiple deletions or recoding or introducing a whole genome, we want still to give ourselves all of the advantages we possibly can. However, I'm very happy to, work with you, and I think it's if we can share the information that would help do something on a small genome, that's fine. Yeah. Can you grow your diatom without photosynthesis?
I haven't in a lab, but there are some reports that you can.
Yeah.
So we have to try it, definitely. And we definitely are inspired with the gene deletion strategy in chloroplast. The transformation method is reported. We are still trying to actually reproduce it in a lab, but the cloning it is very simple now. So that also is very high GC content with about 30%, 32%, I think. But that's the advantage. We can clone it now, but then the delivery is
that's the difficulty. Yeah. Well, good luck.
Thank you. We are inspired by your work.
More than happy to to talk with you as we have been doing. So
I think you addressed all the questions so far. I want to say that was a fantastic talk and I learned a lot.
Good. Yeah, if anybody is interested, there's one more question about why bother producing other molecules in the chloroplast. I think we don't need to recode the whole genome in order to produce those molecules. The advantage of doing it in the chloroplast is that the copy number of the genome means that in principle you can get much higher levels of expression of the proteins, which is important for all sorts of applications. And you don't have to recode the whole genome, but if you want to make neoproteins, then you do have to do that. So,
actually, I have a quick follow-up to that, if you have a moment.
Sure.
Yeah, how similar is the chloroplast translation machinery to bacterial translation machinery? Like, if you were to have, in theory, like an in vitro chloroplast translation machinery, could you take the parts and would they work with each other between bacteria and chloroplasts?
Well, you can take the chloroplast genes and express them in E. coli, and most of the time they're expressed, even using chloroplast promoters and terminators. And you can also express chloroplast genes in E. coli in vitro systems. I'm not certain anybody's done it the other way around, though.
So I think the question is more like, can chloroplast tRNAs be amino acylated and translated by E. coli ribosomes and factors, and can E. coli tRNAs be amino acylated and translated by chloroplast factors?
Yeah.
And ribosomes.
Yeah, so that I don't know if anybody's done that. The in vitro systems used to be, that was the way in which chloroplast genes were, their function was identified using E. coli. Yeah, so I did my postdoc on that several decades ago. And that was before, and everybody here will laugh, but that was before people could sequence genes, and certainly way before anybody could sequence genomes, let alone synthesize them. So that was the way you could just take pieces of chloroplast DNA and introduce them into an E. coli S30, it was called, in vitro system, and then they would be translated. And it was assumed that you were using it was a perfectly normal set of tRNAs and so on. People have tried to introduce bacterial tRNAs into chloroplasts individually, and there's one in particular which is the tRNA for glutamate, and that's involved in heme and chlorophyll biosynthesis as well as protein synthesis. And the bacterial ones, the E. coli ones, don't work. But I'm not certain whether they don't work for protein synthesis as well as heme and chlorophyll synthesis. So.
Okay, because mitochondria have wild translation systems.
Yes, yeah. It's not, it's much more similar to bacteria. It's much more normal, if you like, universal.
Okay.
Yeah.
Yeah, thank you so much.
That's okay.
If there are no more questions.
Oh, sorry. Somebody else has asked me a question. I mean, if anybody needs to go, or if you need to go, Kate, presumably.
I don't, and some people already dropped off, so we can chat.
So Raquel asks about another clammy species that's not well studied. It's considered mixotrophic, but doesn't grow in tap. What would you suggest? Well, good question. We can get ours to grow. We can get Reinhardtii to grow a little bit with glycerol. Chlamydomonas reinhardtii also takes up arginine. We've never tried to grow it on arginine as a carbon source, though. But that's something else you could try. I don't know if that would help, but I don't really know. What species is it that you're growing?
Hi, can I show me her?
Please do.
It's a little bit hard here. Hi, I'm sorry about asking a question so in the end of the seminar.
That's okay.
So I work with Chlamydomonas nivalis, which is the small algae.
Yeah.
But it doesn't which I think it's odd, but it doesn't have a lot of studies, and it's not very well characterized, the species, and it's considered mixotrophic as well. I was able to grow in R2A medium for bacteria that have that have yeast extract, and it's very rich medium, but it grows pink. It doesn't it's not green, and I haven't been able to grow again after that to grow for synthetic.
Do you add any vitamins?
Yeah, I do. I have tried with and without the vitamins, and I also have tried grow in glycerol and acetate without not tap, but just acetate, and it didn't grow very well too. So I would like to. So I would like I'm just like asking a suggestion.
Did you get Nivalis? Was it from a culture collection?
Yeah, UTAX.
They should be able to give you the media that it will grow in, and if it doesn't grow,
I grow photosynthetically in the bold that it's
supposed to grow in.
It's supposed to grow, but I also would like to try to grow properly at heterotrophic to because I studied the resistance with the resistance when exposed to environmental stress. So I would like to see also how it responds at heterotrophic, heterotrophically and autotrophically. So it's just like a curiosity.
Yeah. I'm sorry, I don't have any experience with that. I have a colleague.
No, that's okay.
Who grows
Thank you very
Antarctic
much.
species, but I don't know what he grows it in, but I could ask him if you want.
No, that's okay. Thank you very much.
Okay, no
Bye.
problem.
Your talk was excellent. Thank you
Okay.
so much.
Thank you. Thank you. Good. Yeah.
Thank you.
Yeah.
Any other questions from the remaining?
Yeah. So Gordon, yeah, that's why. Thanks, Gordon. That's why I suggested if Raquel had added vitamins, but she has, so that can't be the explanation.
I'm glad I work with dead things because the living things seem hard to grow.
Not so interesting though.
That's true. That's definitely true.
Good.
Oh, all right, last call for questions. Going once, going twice, and have a great weekend, everyone.
Yeah.
Thank
Bye-bye.
you.
Thank you.
Bye, everyone.